Dear students, today our topic is anatomy of dicot root, and today we'll discuss all the internal structures of a dicot root. Now here we have taken the transverse section of a dicot root, and we will look at different parts, different structures present in a dicot root by looking at this transverse section. Now the outermost layer of the dicot root is the epibellum, or it's also known as epidermis, epibellum or epidermis. Now epidermis or epibellum is the outermost layer of the dicot root, and it is formed of uh, barrel shaped cells, and some of the cells they protrude towards the outer side and form small hair-like structures which are known as root hairs, and these root hairs help in the absorption of the water. Now this is the outermost layer of the dicot root. Now inner to the dicot root, inner to the epidermis of the dicot root, we have another section of cells. We have another part of the dicot root which is known as cortex. It's known as cortex, and this cortex is entirely made up of thin-walled parenchymatous cells. This cortex, it is made up of thin-walled parenchymatous cells, and within this cortex you have parenchymatous cells, and these parenchymatous cells they have large intercellular spaces. You can see here that there are number of intercellular spaces between the parenchymatous cells. Here here you can see large intercellular spaces. So it is the general character of the parenchymatous tissue that the cells are have a thin wall and there are intercellular spaces. So it is same here. So the cortex of the dicot root, it is entirely composed of parenchymatous cells. It is entirely composed of parenchymatous cells. Parenchymatous cells. So you have the epidermis and then you have the cortex. So these are the first two. Epidermis is the outer, and inner to the epidermis is the cortex, and cortex is entirely made up of parenchymatous cells in case of dicot root. Now the innermost layer of the epidermis, the innermost layer of the cortex is the endodermis. This is the endodermis. This is endodermis. It is the innermost layer of the cortex. It's known as endodermis. Now this endodermis has some important properties. If you look at the cells of endodermis are barrel shaped. They are barrel shaped. What is a barrel shaped structure? Now if you look at these cells, you will see a structure like this. They are broader at the center and then narrower at the ends. So they are broader at the center here. These are barrel shaped. So the cells of endodermis, they are barrel shaped. They are barrel shaped. Barrel shaped like this. This is a barrel shaped. And the cells they have a property that on these radial walls, on the radial walls of the these endodermis, for example these are the cells of the endodermis, they have a substance, a deposition of a water resistant or impermeable substance that is known as suberin. So they have suberin deposition on the radial walls, which is a waterproof material, suberin. Now this suberin, it forms a watertight barrier here, for it is present on the endodermis, which is the innermost layer of the cortex, and this suberin deposition, it is a waterproof mechanism. It is a water barrier. It is impermeable to water. It does not allow the water to enter into the central part of the dicot root. However, water has to move. Water has to move from the cortex into the inner tissues. Now, how does that water reach the inner tissues? There are some cells, for example, if we have this is this is the epidermis, sorry, this is the endodermis, and in endodermis there will be cells which will be lacking these cells. Some cells will be lacking this suberin deposition. Some cells will be lacking this suberin deposition, and those cells, they are known as passage cells, because they allow the movement of the water, and all other cells of endodermis will not allow the movement of the water, because they have the suberin deposition. They have a strip like structure. They have the suberin deposition. For example, like the suberin deposition is like this on the radial walls, and some cells will be there that will not have this suberin deposition. Now this suberin deposition is in forms a strip-like structure, and it's known as Casparian. It is known as Casparian strip. It is made up of suberin, which does not allow the movement of water, which is water impermeable. So it does not allow the entry of the water. However, some cells, for example, here this cell, here this cell, here this cell, they do not have this Casparian strip. They do not have suberin depositions. As such, they will allow the entry of water from cortex into the vascular system, vascular tissues, or the central tissues. Now these cells, they are known as passage cells. They are known as passage cells. Now moving inside the endodermis, there is another layer of parenchymatous cells. This layer, which is light pink in color in this image. This layer, these cells, they are parenchymatous. It's a ring of parenchymatous cells, which is inner to endodermis. It is known as pericycle. It is known as pericycle. And this pericycle has two functions. First is that it plays an important role in the development of the lateral roots. So we have a main root, and then we have lateral roots. The lateral roots are formed by the activity of this pericycle. This is the main function of the pericycle. And second, whenever secondary growth takes place in case of roots, dicot roots, then this pericycle has a role in the formation of the vascular cambium, in the formation of the ring of the vascular bundle, that we will be discussing in a moment. Now moving to the inside of the pericycle, what we have is that we have phloem, xylem, phloem, xylem, phloem, xylem, and phloem and xylem. So if you look at the structure, the arrangement of the vascular bundles, these are the vascular tissues, xylem and phloem, is radial. So you have vascular bundles have a radial arrangement. It's like this. This is the circle, and you have here xylem, phloem, xylem, phloem, xylem, phloem. And when we look at the stem and others, you will see collateral conjoint structures. But here you have xylem strand and phloem strand. They are radially arranged, and they are separate from each other. They are not joined together. So here in case of a dicot root, the phloem strands and the xylem strands, they are arranged radially. They are arranged radially, and generally they are 4 in number. They are 4 in number. We have 4 xylem and 4 phloem and 4 xylem. 1, 2, 3, and 4 xylem and 4 phloem strands. Now, the number of these strands, xylem strands and phloem strands, is generally 4, but it cannot exceed 6. It is less than 6 always in case of dicot root. Another important point that you have to remember here is that the condition of xylem is X-arch. The position is X-arch. So you have X-arch condition. X-arch condition. X-arch means that you have metaxylem. These metaxylem, they are large, and you have protoxylem, these small ones. You have metaxylem here, and you have protoxylem here. You have metaxylem here, you have protoxylem here. You have metaxylem here, and you have protoxylem here. So what is X-arch condition? X-arch condition is that you have protoxylem towards the outer side and metaxylem towards the center. This is the X-arch condition. So here you have to remember three points, that the arrangement of the xylem and phloem strands or the vascular bundles is radial in case of a dicot root. Number second, their number is generally 4, but it can be more, but it cannot exceed 6. It is less than 6. And the condition or the position of the xylem is X-arch. You have protoxylem towards the outer side and metaxylem towards the inner side. Third important point. Now you have, this is phloem, and this is phloem, this is xylem, and between xylem and phloem there are cells. These cells here, the cells here, parenchymal cells here. These cells, this is known as conjunctive tissue. It is known as conjunctive tissue. Conjunctive tissue. So the xylem and phloem, there is space between xylem and phloem which is occupied by cells, thin walled cells, and this tissue is known as conjunctive tissue. Now in the center there are large parenchymal cells, and this central part is known as pith. It is known as pith, and you have to remember, pith is not well developed in case of dicot roots, while as in case of monocot roots it is very well developed. Now we will look at how secondary growth takes place, how secondary growth is possible in a dicot root. Let's see. Now during secondary growth, what happens? The cells above this xylem strand and below the phloem, above xylem, below phloem, above xylem, below phloem, above the xylem, below the phloem. All these cells, they become meristematic. So the cells which are present below the phloem, and these will be the cells of this conjunctive tissue, and above phloem, which will be the cells of the pericycle, they become meristematic, and then they join together. They join together like this. As they join, they form a ring-like structure. They form a ring-like structure, and this mirrors, it's known as cambium. Now these cells, they will form new cells towards the inner side. They will form new cells towards the inner side, and the xylem, which is present towards the inner side of the ring will be pushed towards the inner side, and new xylem will be formed. This will be the primary xylem, and the new cells will form new xylem, that will be the secondary xylem. No, outside this tissue, when this cambium, it will form cells towards the outside as well, and this cambium, it will be pushed towards the outer side. It will be now secondary xylem, and new xylem will be formed as well. New phloem will be formed on the outside, new xylem will be formed on the inner side. So this ring, as it forms new cells, it will become somewhat oval in shape. So this ring of this cambium, it will cut cells towards the outer side, it will cut cells towards the inner side, leading the formation of secondary xylem and secondary phloem, and it will increase in the girth of the root. And this increase in the girth by the activity of this vascular cambium, by the ring of this cambium, is known as the secondary growth. This is how secondary growth takes place in dicot root, and we will also be discussing the secondary growth in case of dicots in a bit detail in the coming lectures. Right now we have to focus on the anatomy of the dicot root. So anatomically, if you look at the dicot root, you have the outermost layer, that's the epidermis. And some cells of the epidermis may protrude out from root hairs. Now inside to the epidermis, you have a region of parenchymatous cells with large intercellular spaces. The cells are thin walled, it's completely parenchymatous, and it's known as cortex. Then the innermost layer of the cortex is the endodermis, which are characterized by the presence of the Casparian citrate, which is a water impermeable material made up of suberin. Now some cells in case of endodermis do not have this Casparian strip, they are known as Passé cells. And then inside this we have another layer which is known as pericycle, which plays important role in secondary growth and also in the development of the lateral roots. And inner to this you have radially arranged vascular bundles. We have 4 xylem and 4 phloem bundles, and the number of radially arranged vascular bundles in case of dicot stem cannot exceed 4. Now between this xylem and phloem strands you have the conjunctive tissue, and we have centrally located pith. This is the general structure of a dicot root. Thank you very much for having me here.